Okay, here today, our group going to present the derivation of signal to noise ratio for the filter back projection. So first, I would like to briefly introduce the CT noise and filter back projection. In principle, the CT image noise can be classified into quantum noise and detector noise. The only noise we are going to talking about today is the quantum noise, which is produced by the stochastic fluctuations regarding scattering and absorption while the radiation passes through the object. Filter back projection, as we learned from the lecture, is the reconstruction algorithm that can reduce those kind of quantum noise in the CT imaging. And filter back projection is a fast and direct method, and as the name suggested, it will have two main steps: filtering the data and performing the back projection operation. And more about quantum noise. In other words, it is a random process due to fluctuations in the number of photons regarding the detectors from point to point, reaching the detector from point to point. And quantum noise has an undesirable effect, mainly in imaging using low radiation dose, because there's a trade-off between the dose and the imaging acquisition. With the low dose imaging, the imaging will be noisier, but high dose imaging will give us a better imaging, but but also will cause increase the radiation, future radiation risks for the patient. Therefore, for each specific imaging, it is important to choose the appropriate radiation dose so that we have a reasonable signal to noise ratio, which is the noise is not too high and the signal can be visualized. The last concept I want to introduce is the Poisson distribution. In theory, Poisson distribution is the probability distribution of events over a fixed number of time or space for independent events. The magnitude of the fluctuation for the quantum noise is given by the square root of the number of photons contributing to the measurement. And the number of those photons reaching the individual pixel on the detector follow this Poisson distribution. The graph here shows the Poisson distribution, and as you can see, for the high dose imaging, the photons collected by the detector will follow the Gaussian distribution. But when we have low dose imaging, which we have less photon, the minimum number of photons will have the highest possibility to be collected by the detector. Due to the limited number of photons, the low dose CT imaging is usually simulated with Poisson distribution.
Okay. Before we go to simulations and the model we use, here I just briefly go through the mathematical derivation, the key steps we use involving our models. So there are two key assumptions we made. The first one is the electron has independent probability, especially when hitting the target atom for the photon emission. The second one is ideal detectors. So that means every photon it encountered, it will count it efficiently. So starting from the n star, which is the expectation value of the total amount of the capital N with the individual probability of P, we can write down the binomial model as equation 2. And here the P is equal to n star over n. And therefore, when n is very large, we can further simplify this probability of a certain amount of photons get emitted from the target into this equation 4 here, which is a Poisson distribution. And notice when the n is very large, by starting approximations, this mathematical model can be simplified to the Gaussian noise distribution. Okay, so here is the signal to noise ratio equations, which is mean over the standard deviations. For the mean, you just plug into the probability equations we have in the slides before. And I write down the detailed mathematical equations on the right-hand side. Just try to expand the terms in before the E portions. And for the variance, essentially it's the same way for calculation. You just need to expand the contents inside the square into the pretty complicated, a little bit more complicated portion and do the simplification. And basically the SNR can be the root square of n multiplied with probability P, and with expanding the N, you get should be a square root change of the tube current. The T here, the capital T, is the time. We put in our model, which is usually 1 millisecond here. And how we apply this mathematical model into our projection simulation is that we use the projection initially, the Raygun transform of the object as projection to take it as a possibility of the photon get transferred to our detector. And then we multiply it with different tube current. By that, we can further have the Poisson random functions out of this whole multiplies. And after that, because the result we get is the total is similar to the total photon output. So then we normalize it again by tube current. And therefore we can have this noise processed image for the future. And the two key assumptions we made here is besides detectors before, the second photon are either transmitted or removed in this process, which is also pretty important.
So in order to prove our concepts, which is the signal-to-noise ratio is proportional to the square root of the dosage, we modified the MATLAB program for the homework 3. So, at the first, we created a hedge Logan phantom on a 400 × 400 space, which means it's 0.5 millimeter for each pixel length, and it is the top left image. And then we run the radon transformation to get the GL theta. The theta is ranging from 0 to 180 degrees with a delta theta equal to 0.5 degree. So we get this noise-free GL theta. Then we use the concepts we have talked about to generate the random number from the Poisson distribution to add a noise on the GL theta, which we get the Poisson noise added on the signalgram on the top on the bottom right image. And the idea is at a certain projection angle and the linear displacement, the signal received by the detector has a Poisson distributed noise controlled by the magnitude of the tube currents. So in this case, in 10 milliamps as the tube current inputs. Then we use the inverse Radon transformation with a Hamming filter. So we can still see the edge here, and also we can see some like pattern inside the Logan phantom, and but it's really noisy. So in order to prove our concept, we input 100 different tube currents as a source for our systems. Then we calculate the differential signal-to-noise ratios by picking two regions. The top red box, which is the homogeneous region from the phantoms, and the bottom right red box is another region only contains the noise. The differential signal-to-noise ratio is equal to mean difference of the signal and noise, and then we divide it by the standard deviation of the noise. So the tube current ranges from 0.1 milliamps to 1,000 milliamps. So usually the smallest tube current is 10 milliamps. So we can observe on this pattern. So we fitted the power fit to our data, and we get the signal-to-noise ratio is equal to 0.1 times the tube current to 0.49. So which prove our previous concept. The signal-to-noise ratio is proportional to the square root of the tube current. And in our fit, the R square is 0.98, which means that is a really good fit. And in summary, the imaging reconstruction methods, the filter back projection, can increase the signal-to-noise ratio and improve the image quality. And the signal-to-noise ratio is proportional to the square root of the radiation dosage. It means the capability of improving the signal-to-noise ratio outcome gradually decreases with the increasing radiation dosage. And an optimized radiation dosage can be determined to minimize the risks for the patients, but also acquire a relatively good quality image. And here are our reference. And thank you so much for listening. Do you guys have any questions?
Great, thanks. Nice talk. Nice project. Any questions from the students? Did you consider looking at a phantom where you had a really bright object, like a very intensely absorbing object, and see what it did at the low MA? That's sometimes
So in our this quantum, I think the edge is really bright.
Yeah.
Correct. And so we can still see the edge even with the Poisson distributed noise. So we should definitely think about that and maybe run some other experiments on it.
Because it's those bright objects that reduce the photons down to the level where the Poisson distribution really matters versus a normal distribution. So it's interesting.
Yes.
That makes sense.
Any other questions? Okay, terrific. Thanks so much.
Thank you.
Okay, I think we're next. Can everyone see my screen?
Yes, we do, but I think it's not a full screen. If you can rotate with the wide angle, it'll be great.
Okay, give me a second.
Yes,
that better?
yeah, perfect. Yeah, thank you.
No problem. Okay, so hi everyone. We did our project on the impact of filters on spatial resolution. I'm Rebecca and I work.
Turn up my volume. We were getting horrendous feedback with that, but okay, let's try that now.
Is it okay now?
Oh, good. Thanks.
Okay. All right. I'm Rebecca and I worked with Maria and Isaiah. Okay, so to start, we have some background information on filter back projections. To reconstruct the image, the Fourier transform of the projection is multiplied by a filter, which alters the projection data. The image is then inverse Fourier transform and then smeared back or back projected across the angle the image was taken at. Each of these back projections are summed to make the reconstruction of the image. And this is shown here in the image to the right, where the reconstructions at different angles are summed. Typically, the more projection angles increases the signal to noise ratio and therefore image quality. A filter back projection is better than a simple back projection with no filter. The filters help to remove blurring, therefore increasing resolution and making an overall better reconstruction. Okay, so there are different types of filters used in the filtered back projection method. To create the filter, different kernels are multiplied by the ramp function, which we'll show in the following slide. The filters are used to filter out low frequencies that cause blurring in the image. This results in a sharper image that has more clinical relevance when examining small objects. Different reconstruction kernels are used depending on the image that will be taken, such as if it will be bone or tissue, which have different attenuation coefficients. There's a trade-off between noise and sharpness in the final image when selecting a filter. And the image on this slide shows reconstruction images using different kernels. It's likely that the ones on the left here are smooth kernels such as cosine or Hanning, and the ones on the right here are sharper kernels such as Shep-Logan or Ramlack. For the smooth kernels, you can expect the signal to noise ratio to be better, while the sharp kernels look grainier, meaning the signal to noise ratio is lower, but the resolution is typically greater. Okay, so these are the different kernels that are used in filter back projection. We made these in MATLAB. So the base filter is this here, the Ramlack filter, which is known as the ramp filter. Each of the other filters are made by multiplying a window or function by this Ramlack filter. The Shep-Logan here is the ramp filter multiplied by a sinc function. The cosine filter is the cosine function multiplied by the Ramlack. The Hanning is the Hanning window multiplied by the Ramlack. Sorry, this is the Hanning. And then the Hamming is the Hamming window multiplied by the Ramlack. So some of the filters are sharper, such as here with the Shep-Logan, and some are smoother, such as the cosine, and this generally leads to different signal to noise ratios and resolution in the final images.
So resolution is the degree of blurring that a medical imaging system has towards a single point in space. When looking at two separate points in space, the less blurring that a system introduces, the closer the 2 points are, and can still be distinguished as two separate points. The best medical imaging system should have low blurring to have the highest resolution. One way to measure resolution is the full width at half maximum value, which is the minimum distance that 2 points must be separated in space in order to appear separate. This is seen in the image on the right. As the points travel closer together, the full width at half maximum value decreases, which results in an increase in resolution. The dips in the separate images are important in seeing whether the 2 points will be visualized as separate. Another way to measure the resolution is using the modulation transfer function, which is the smallest separation value between two adjacent maxima or minima, and a sinusoidal input that can be resolved in the image. In our project, we will use the full width at half maximum value to determine best resolution with the kernel filters. So for our project, we created an image in MATLAB with two impulses that move closer together until they are at the same position as seen in the first picture on the left. We created a sinogram of this image with Gaussian background noise as seen in the third image, and then took the inverse radon projection, which is the filtered back projection. We then took the profiles through every pair of pixels for each filter, and then determined at what distance we were unable to differentiate the two different impulses. We also examined the signal-to-noise ratio. We then concluded which filter provided the best resolution.
Using MATLAB's IM profile function through every pair, we had a total of 48 profiles through the pairs of pixels from the 6 filters. The images on the bottom of the slide represent the profile through the pixels where we're able to determine the 2 separate impulses. The 2 larger images are representative of the difference between 2 distances, where we define distance as the number of pixels in between the pulses that use the same filter. In this case, it was the cosine filter. In the distance of 2, we could see that the impulses are indistinguishable from each other. And while in the distance of 3, we can clearly see the 2 peaks from the impulses. This is the idea which is the focus of this project. One of the first things that we notice when looking at the titles is that the distance between the pixels are not the same in every case. For example, when using the Ramlack and the Shep-Logan filter, we could see that the distance of 2 pixels, we were still able to differentiate between the impulses because of the dip seen in the middle. However, this dip was not apparent in the cosine, Hamming, and hand filtered back projections at the distance of 2, and instead were less recognizable when they were 3 pixels apart. Thinking back to our earlier slides, we can see that there's a connection between the reconstruction filter type and resolution. Both the Ramlack and Shep-Logan have the sharp edges, which other filters for the most part have smoother shapes, which cause blurring. So although signal to noise ratio is different from resolution, we examined signal to noise ratio to get a general idea of the quality of images which is produced. In this specific case, we found that the images were all quite similar with a signal to noise ratio of about 1. The only major outlier was the naive back projection, which used no filter, which when thinking back to our course and background information, we expected as no filter tends to lead to extreme blurring. However, due to the images that we used, which did not contain different attenuations, edges, and shapes, these results can only really tell us that the filtered back projection was useful more clinically than non-filtered back projection. We could not draw any conclusions in terms of this signal to noise ratio between the back projection filters and use it as a general metric for the filtered versus unfiltered. In our conclusion, Maria will expand on the general signal to noise ratio expectations with these filters. However, comparing signal to noise is out of the scope for this project between the filters.
In conclusion, we determined that the filter with the highest resolution of the impulses is the Ramlack filter. We can see with the images on the right the difference in the image resolution when there is no filter and when using the Ramlack filter. According to a paper on the effect of reconstruction parameters on noise and resolution, the Ramlack filter usually gives the highest noise levels and spatial resolution because the Ramlack filter emphasizes high frequencies, which makes it sensitive to noise but also more responsive to edges. Also, in general, the more rounded the filter graph is, the higher signal to noise ratio that it has, as it increases blurring, which typically reduces noise. This project is clinically relevant and important because CT scans need high-resolution images to tell the difference between small objects in the body and know which filters are the best to use to create images with high resolution and high signal-to-noise ratio. These filters are commonly used to differentiate objects in images of soft tissue and bone. Moreover, studies also show the importance of the use of kernel filters in CT screening on patients with lung cancer, emphysema, and other diseases to detect the small phenotypic changes on the tissue. So that's it for our project. Thank you for listening, and we will take any questions now.
Terrific. Thanks very much. Any questions from the students on the project? I have a question. That was the signal to noise table you pulled up there looked surprisingly constant, right, in terms of signal to noise. And this is the signal to noise between your single pixel dot and the background. Is that correct?
Yeah, that's correct.
Right. So it's an interesting question, and that is sometimes signal to noise depends on what it is you're imaging. And when you're imaging a dot, these things might produce a measurement that looks like the signal to noise isn't changing. However, if you wanted to image something the size of a dime, right, and so it had quite a bit of area that you could integrate over, then your signal to noise probably would be quite different with these filters, I think. So it's a deeper question, and it's quite interesting. But the fact that there's constant, right, with these filters is surprising, but I think it's the fact you're imaging a dot.
Yeah, exactly. That's why we mentioned that we didn't use an image with different attenuations, edges, and shapes, because typically speaking, something like the handing or the cosine would have a higher signal to noise ratio.
Right. Great. Okay, terrific. Thanks. Good simulation. Okay. Who's coming next? All right. Sam Duke, Mike Teller, and Naomi Wilcox. Is that all right? Great.
No, we talked about this. We changed our topic. I don't think I actually updated it on this spreadsheet.
Oh, okay.
Yeah, we're the low energy CTs.
Okay.
I talked to you about this.
Oh, okay. So, well, bring it on up and let's see the title. Yeah, I think you need to share the screen on the. You should be able, are you guys on the network? You should be able just to share the screen and it'll come up here inside, right? All right, so just go to Zoom screen share thing.
Has the previous team stopped sharing? Yes, okay. You're on the network. You can just join the Zoom call. Oh, you haven't joined the Zoom call? Yeah, better do that first. It's not that one. Perfect. All right, excellent. Here we go.
Okay, so our presentation is on weighing the deleterious effects of radiation dosage on a patient population against the need for sufficient energy and imaging. So I'm going to begin with the biology of radiation. So we talked in this class explicitly about how radiation is bad. Obviously, dosage is something we have to take into consideration when doing CT imaging. But in what ways is it bad for the patient, or what does the actual effects look like? So to first begin with that, we have to characterize ionizing radiation, which we briefly touched on in the book in the class. That's just defined as any radiation with sufficient energy to damage or alter DNA in many different ways. They have primary effects, which is the most deleterious, which is double strand breaking, or as they sometimes shorten to DSB. And then we have secondary effects, which is oxidative damage. When your body incurs a double banded strand break, what we have is two possible ways in order to fix that. We have non-homologous end joining at HEJ, as well as homologous recombination HR. So predominantly in eukaryotic cells, such as us mammals, whenever we endure a double-stranded break, we go through non-homologous end joining. Even though that is predominant, it's easiest, it's low fidelity, and has a high risk for mutations and kernel mutations. Homologous recombination, you might think, well, why don't we go through that? However, if you notice, in order to go through homologous recombination, it's a much more involved process that requires a sister chromatid. Therefore, it can only take place during the S cycle and the G2 cell cycle during phase replication. So since we normally don't have the sister chromatid around in order to share the DNA, we just have to kind of paste the two things together and hope they don't work. On the other hand, as I said before, we have oxidative stress. Oxidative stress can lead to similar problems such as double strand breaking, changings of bases. Bases are the individual, the smaller component that kind of cross-links the DNA helix. However, basically from a 0.2 milligray radiation exposure, which is in line with some CT scans, they say they approximate about 600 or 6,000 base changes. However, on a daily just exposure radiation, you go through about 30,000 base changes. So that's well within the range of what your body can handle in terms of DNA repair. So the primary concern is the double strand breaking. Now going up a little bit more from a pure gene to a molecular view, it's not updated.
Is it updating on your computer but not on the call? Why would it not be updating? Says you are viewing Naomi Wilcox's screen. Options. There you go. Okay.
Okay. So taking a little bit wider approach, now we're going to look at a genomic view of radiation and how it affects you. So we have an idea of an exome. An exome is defined as the part of your genome that codes directly for proteins. So in other words, it's everything of these exons. You have exons and introns. Introns, on the other hand, are non-coding regions. Generally, your entire genome is about 1% coding regions to 98% non-coding regions. Okay, and obviously the 1% is because some people say 1%, some people say 2%, so that's why it's 198. I know it doesn't add up to 100. But the non-coding regions, even though you might think they're throwing away because we don't have any bit of proteins, those are very important in terms of regulation of gene expression, of determining when genes turn on and when they turn off, which in turn has a large effect on cancer and tumor genesis. However, with the exons in the exome, our first point, those variations and mutations in the exome are attributed to 85% of disease traits that are of large effect. So in other words, 85% of pretty deleterious diseases occur in single or only a few small genes within your exome. So generally those are considered even worse. So if we go back from thinking to the previous slide, if we have a double strand break and our DNA is disturbed within the exome, we have a very high chance of a serious mutation, serious disease, whereas a much lower chance occurs in non-coding regions, even though those predominantly make up most of our genome. And then even a little bit more, so in terms of your body, you have somatic and you have germ lines. Now somatic, don't worry about the writing, this is just for color representation. Somatic is pretty much everything outside of your reproductive genes, your gametes. So that'd be like your skin cells, your arms, your hair, right? So within your somatic cells, quiescent cells, in other words, slow dividing cells, are less radiosensitive. So your nervous system, as we know, tends to divide slower, hence why you can't really recover from nerve damage later in age. However, your hair, your skin, your nails, your epithelial cells, and your gastrointestinal tract, those divide quickly. Those have a higher risk of mutation because the more you divide, the more mutation you can occur. And then the germline cells, we have oocytes and spermatocytes, which is pretty much eggs, sperm. Those have a very high risk of mutation because they tend to divide more, especially sperm cells, which carry an extreme risk because of this extreme number of divisions they occur in their lifetime. So this all comes, in fact, going from the last slide, because when we have our gametes and we have our spermatocytes or our oocytes, we're talking about inheritance, right? We're talking about inheritance of our genetic factors the same way you inherit your eye colors, your height, your weight, other genetic factors. You do have a risk of inherited mutations. The reason why I leave a question mark here is because weirdly the science behind radiation-induced and genetic inheritance is kind of a toss-up. They have studies back in 2005 looking at irradiated mice and the prevalence of genomic aberrations in their offspring. They found a very large significance increase in over 100,000 kilobase pairs deletions or inversions, which are generally considered the most deleterious, both in frequency and in terms of spectrum of where they occur. So, however, in a very real-world explanation of this, back in just April of this year, 2021, they published a study saying, looking at the children of people that were exposed to high levels of radiation during Chernobyl, and they found no significant changes in genes or no more prevalent deleterious effects or mutants arising from that single extreme dose of radiation, far greater than anything we see from a CT scan. So the reason is, it's kind of a tossed bag whether we're going to have these inherited mutation risks or we're not. Now, on top of the inherited mutation risk, we have individual genetic risk. So in a study of 402 breast cancer patients, they found that only 30% of the variation in terms of individuals' cellular response to acute tissue damage from radiation could be contributed to, clinical factors such as, hemoglobin levels, serum calcium, as well as, their hypertension and smoking habits. Only 30% could be attributed to those factors. Therefore, what they conclude is there must be other epigenetic factors, other factors in essence, where the environment dictated the individual response and their own genetic makeup determine how much radiation damage their own cells endure. And then last, to leave off, another thing we have to consider from an organism view is evolutionary pressure versus youth. So how evolution worked is it pushed us to, as a species, as with many other mammalian creatures that have to undergo, because we're larger, a lot of cellular divisions, they suppress tumors early in growth. However, those controls on tumor growth early in life tend to fade away later in life because you presumably had enough time in order to reproduce and pass forward your genetic information. However, that comes into play when we're taking into account CT doses in children, that as a child with more of their life and ergo more mutations, time to have mutations and more opportunities to grow physically and more cells, we have an inherent risk of what began as a small mutation eventually accumulating into a large, very serious effects.
All right, so going from the micro level to the macro public health and population scale, this is a slide I think we've seen in lecture, and it's from the New England Journal of Medicine paper. And the data is slightly older, so it ends at about 2007, but the point here is the trend. So this is sort of shows an exponential increase in the number of CT scans performed every year in the US. So currently, over 80 million CT scans are performed in the US every year, compared with just 3 million in 1980, so a big sharp increase. We think this increase is just mainly due to the advanced technology and the fact that it's widely available. As Michael was saying, we have higher risk groups. So in CT, we're really concerned with the fact that children are not only more sensitive to radiation, but they have longer life expectancy, so they have more time to accumulate risk. And as well, women have been found to have a higher risk of developing certain cancers than men, comparing men and women of the same age and the same dosage. So here's just a brief chart from the American College of Radiology. And some certain areas of the body can have higher effective radiation dose risk. So here we have by area, we have the millisieverts, so the effective dose, and then compared with the natural background radiation exposure that it would take to have an equivalent dosage. So we can see that some areas of the body have only days of radiation exposure, and others have 5+ years. So as I said, CTs have have been linked to greater life expectancy and declining death rates. But the American College of Radiology does say that there must be a clear medical benefit that outweighs the risk of taking on this dosage. And they have a concept known as the as low as reasonably achievable concept, which just means we should have the minimum dose possible to the patient. So some drawbacks versus benefits. You know, ionizing radiation can increase the risk of cancer. So that's a drawback. We also have, there's been known to be some allergic reactions to contrast agents such as iodine, but the benefits we think are very large. So it's non-invasive, painless, and it's high resolution. You can image multiple tissues at once, and you can avoid missing a diagnosis in certain patients that can lead to further complications down the line.
All right, so how can we reduce these, how can we minimize the radiation dose to patients? So there are two main approaches that we could consider. The first one is reducing the sample rate, so the amount of times that the CT machine takes a picture as it rotates around the patient. The second is to reduce the intensity of the beam, so lower the number of photons that pass through the patient, limit the number of chances for DNA to be damaged. And so we created a simulation in MATLAB used based on the MATLAB code we used for homework 3. This is the phantom we used. We see we have 4 squares of varying brightnesses, and the lower graph is a slice through the phantom of the brightest and the darkest square, so you can see exactly what the result of these techniques are. So the first technique is reducing the sampling rate. So as mentioned, this is taking less pictures as you go around, so the patient is exposed to less radiation. This affects the delta theta of the machine. Most commercial CTs have a delta theta of between 0.25 and 0.5. And as you can see, those actually do look pretty good. But as delta theta gets bigger, you can see there's an increase in artifacts due to aliasing and back projection. Whereas as you get to a delta theta of 5, it's just completely distorted, and there are artifacts all over the place. You can see in the cross section, it's very heavily distorted. So that's probably too much undersampling. So then the second thing is reducing the beam intensity. This lowers the signal, but also means that there's a decreased signal to noise ratio. And so you can see the noise is increased as we go up. And this will obviously depend on a lot of factors, what the type of machine, the type of patient, and various other effects. And this is particularly tuned to each specific scenario, which is part of the art of taking CTs. But as you can see, there is a limit to how you can do it. All right, that is our presentation.
Great. Okay, any questions from the students? Okay, I think we're running a bit behind time. It was a long presentation, so we'll just move to the next presentation. Thanks, guys, for that simulation. Practically speaking, now I have a very good understanding of whether or not you want to get a CT scan at your current age. When you go to the hospital, you can make your own ALARA decision. Next up, we have sensitivity versus filtering. Okay, great. So you guys can take the screen. Are you on the call?
Yeah.
Okay, terrific. Okay.
Hello. All right. Good morning, and my name is Don. This is Carol. This is Mina, and our topic is on filters that can be utilized in CT scan to show the characteristics of collocation. And there are some typical features that can be seen in collocation. Features are generally shown bilateral and in peripheral region of the lung. And specific feature like ground glass opacity, which we're going to call GGO later on in our presentation, will be shown on the picture at 8. And in the middle stage, GGO with visible intraocular called crazy paving. And later on, the stage will be shown as the consolidation and maybe cavitating lesion. And abnormal activity are usually on the peripheral area of the lungs, we can see in this study. And comparing to the patient with non-COVID-19 pneumonia, most of their abnormal activity on the central area of the lung. Therefore, in our project, we can have a focus area on the image to improve our filter, and the filter can be better emphasize the COVID-19 CT image feature. And one of the characteristics of them being is that they only allow through radiation, which is the primary radiation that has passed through, has to be attenuated by the patient to reach the detector. And this signal is going to be part of the reconstruction kernel. In the helical CT scanner, the X-ray tube and the detector continuously rotate around a large circle around the patient, which generate many images from different angles. And according to our textbooks, for the helical scanner, the continuous movement of the patient to projections cannot correspond to the same point. Therefore, the fan beam projection is identified by its longitude position Z subscript J, measured by relative to the radiation. And the index J, in fact, not only identifies the position C subscript J of the projection, but also determines angle. And many single size image will be generated through this process while the X-ray tube spins around the patient. And the image acquired through fan beam CT scanner have superior spatial and contrast resolution. The X-ray attenuation of the energy used by CT is positively correlated with the density of the tissue and an organ in the human body, because subjects of the human body have different atomic numbers and information. Therefore, the detecting attenuation of X-ray, we can reduce the density of different locations in the human body through multi-step conversion computer and image reconstruction.
Okay, so here's the, oh, sorry. Okay. So this is kind of like the typical CT scan image for COVID patients. So what we're gonna do is to figure out how all the above features reflected in the sizes of the CT image. So the first one is kind of like the most important features in the COVID patient, the ground glass opacity, which appears as the hazy gray areas that can show up in the CT scans. So it is kind of like the air spaces are becoming partially filled with the fluid gel and inflammatory cells, etc. So here is the reverse halo, which is the focal rounded area of the ground glass opacity, surrounded by the complete or near complete ring of the consolidation. And the central area corresponds to the alveolar septal inflammation and cellular debris in alveolar spaces, while the ring-shaped parallel air spaces consolidation corresponds to the granulomatous tissue within the distal air spaces. So here is the crazy paving feature, shows up as a linear pattern, which can occur when the spaces between the lobule widen. And this is also called the consolidation, refers to the alveolar air spaces being filled with the fluid, inflammatory cell tissue, or the other materials. During the consolidation, it will generate it while moving to the late stage of the COVID. So as we find so many features in the COVID patient CT, the next step is what we want to do is to amplify these features and to make it more clear and easier to be observed, in which we apply the different features to these images. So the filtering is a technique for enhancing the quality of the image by reducing the noise in low dose radiation CT. In such a process, image processing operation implemented with the filtering include smoothing, sharpening, and edge enhancement. So these are the images processed by the Toshiba CT scanner, and we cannot apply the general features we learned in class to these images in the case. However, it doesn't work so well. So we find a new different filter which can be applied in our cases, the silver filter and the smooth filter. So before we move on to new filters, I want to talk a little bit about why the previous four filters doesn't work well. We think because the modern CT scanner already did a good job in filtering and back projection. So repeating the process could involve the more noises when we convert the image back to the spatial domain. So that's why we started to look for new filters. So the first filter we used is silver filter, which is commonly used in detecting the edge of CT scan. So for grayscale images, edge is where it has a strong changes in brightness. This filter comprises the two 3x3 kernel shows in the left picture. The kernel on the top and bottom calculate the gradients for each pixels for detecting edges in X direction and in Y direction. If we take a closer look at the matrix on the top, it has a 0 column vectors in the middle and 1 positive and negative column vectors on the left and right. So when applying this filter, it emphasizes the brightness difference between the left and right, which means we can see the edges when we move in the X direction. And same thing for the Y kernel. And we apply the super filter to have a better observation of a reverse halo. And here is the original image of the reverse halo. And this is for when we apply for it. And we also use the silver filter for have a better look for the precipitation. Those are the original. The pictures on the right is the precipitation. And when we apply the filter, you can see the middle one is the precipitation. Sometimes it's hard to tell the difference between GGO and the precipitation in the original pictures. But when we apply this filter, we can tell the difference like between them clearly. Because for the precipitation, there's a honeycomb structure in the precipitation area, but for the GGO, it's hard to tell something like that. So the next filter, as we mentioned in the previous slide, the silver filter can detect the edge, which has a high contrast very well. But for GGO, which has a blurry edge, it could be hard to see. So in this case, we use the mean filter to smooth the GGO. Mean filter is one of the simplest low pass filter. When we apply for it, each pixel and its 8 neighbors are multiplied by 1/8 and added together. So the pixel in the center depends on its neighbors. As an example, on the picture on the right, the vessels and the bronchial structures may still be seen in the GGO. And when we apply this mean filter, it filters out the projection of them. So it makes the GGO more obvious to see. Here is the original picture. The GGO picture is on the right. And this is when we filter it out. It could be a little bit hard to see from the slide, but we can zoom in. It's much better to tell the difference. There is one more filter called bubble filter. When we were looking at some research paper, we saw researchers use it for texture analysis. But we had trouble applying this filter to our CT images. Probably because we don't have enough knowledge or to understand the texture, especially to recognize the texture from the output result of the filter. And all of the 3 filters we mentioned in the presentation have been used in machine learning for future COVID-19 detection. So here's all the references we use in our paper, and thanks for listening. So any questions on that? Oh, sorry.
That's okay.
Yeah,
it stopped updating. Yeah, that's like, okay, good. It's just a list of references. Okay, terrific. Thanks very much. Good. Any questions for the group? Yeah, from the back.
How did you download the images? Were they available from the papers or?
Some of them are from the papers, but we had download from this database that it's a DCIM format, and then we use the DCIM like an app to like open it up, so it has like all the information.
Okay, so you took the DICOM images.
Oh no, we downloaded the picture as a TIFF format, and then we applied filter.
So when you're looking at different CT filters, was that done by the author?
Oh, we applied the filter.
For the previous few slides, when we introduced the features of the COVID patient, that is from a paper. But for the later, when you see like the comparison between the original and the filtered image, that's what we did in MATLAB.
So the different CT filters on the COVID patient, that was in the paper.
Oh, when you had the 4 different filters, the classic CTVs.
We did it. Yeah, we did it ourselves. Yeah.
Okay, so that's you downloaded the TIFF and then for DICOM, or?
No, DICOM converted to TIFF and then MATLAB, and then we filtered out. Yeah.
And it's interesting that those filters gave the same result, right? But so what would be also interesting is to look at the, what does a say a Sobel filter look like in Fourier space?
Yeah.
Right. And then what is it enhancing? And it's obviously enhancing edges, right? That's what it looks like it's doing. But it would be interesting to also plot that and say, here's our original MTF of this CT scanner using the Ramlock or the Hanning, and then when you do a Sobel filter, what does that do in the Fourier space as well? So, okay, great. Thanks, guys.
Thank you.
Thanks. All right. Spatial resolution versus radial position with Chloe and Derek. Fantastic.
All right, hi, my name is Chloe and my project partner is Derek. We're doing spatial resolution of CT as a function of radial position as our project.
Very good.
Okay, to start, it's important to define some key concepts and background. As we all know, spatial resolution is the ability to distinguish between nearby structures. When two structures are close in proximity, high spatial resolution is required to differentiate between them, whereas low spatial resolution may blur them into one structure. Spatial resolution increases with decreasing pixel size and decreasing field of view. It's also important to understand radial position, which is the coordinate system that we'll be using throughout this project. Radial position describes a point along a radius from the isocenter of the object. The modulation transfer function describes how an imaging system attenuates higher spatial frequencies. Depending on which signals are allowed to pass through, the modulation transfer function characterizes the effect of the imaging system on spatial resolution. As you can see in the figure here, the lower spatial frequencies with just a few lines per millimeter can be easily distinguished in the image, but the higher spatial frequencies with many lines per millimeter are attenuated and no longer distinct. Because the MTF is the Fourier transform of the point spread function, it serves as a useful way to quantify spatial resolution. Many CT images have artifacts and radial streaks, which is due to undersampling in the theta direction. As you increase the number of views, you approach adequate sampling as defined by the Nyquist sampling theorem and a better signal-to-noise ratio, which results in a more realistic image of the object. In order to understand the field of view in which you do have adequate sampling, we can consider view aliasing. This concept describes how overlapping projections cause a buildup of sampling in the center of the image and a lack of sampling in the periphery of an image. In other words, view aliasing explains why the edges of an image do not have as high of a spatial resolution at the middle of an image. Here, we can visualize the concepts that I just discussed on the previous slide with a real CT image of a heart. We created this simulation that generates the inverse radon transform with the Hamming filter at various sampling angles from 0 to 180. As we cycle through the delta theta values, we can observe how spatial resolution is dependent on the view count. There is always higher fidelity in the center of the image where there is an adequate sampling rate when compared to the edges, but this safe zone gradually diminishes as the sampling rate decreases. Notably, at very low sampling rates, we observe radial streaks forming near the edges of the images. At lower view counts, we also observe worse signal-to-noise ratios.
All right, so let's go a little bit into the physics of why this occurs. This diagram shows essentially that we have two different regions here. We have one region in the middle where we have where the points in the theta direction are adequately sampled. And that's because the value of delta S, which is the theta displacement, is smaller than the delta L, which is the radial displacement. And anywhere beyond this green circle that we sort of denoted as the safe zone, we experience aliasing, and that's due to the points in the delta S direction or that's due to delta S values being greater than delta L values. And the diameter of this safe zone can actually be calculated using the number of views. And in our simulation, we use a number of views of 90 with a delta theta of 2 degrees. And that helps us identify the size of the safe zone when we later characterize the radial position function. Okay, so now how can we generate an actual function that describes spatial resolution versus radial position? So the approach that we took here was to generate two diagonal lines, and we were increasing the sort of the pixel separation that occurs between these two lines. And going off of the definition of spatial resolution, if after we take the inverse radon transform of this image and we're still able to resolve that there's two distinct lines, then that means that the spatial resolution at that point is greater than or equal to the amount of pixels that are separating the two lines in the original image. So if we have a look at this the simulation here, we are gradually increasing the pixels between the two lines. And after we take the inverse radon transform, you can see this point that's gradually going outwards in the radial direction. That's sort of giving us the coordinates at which the spatial resolution is equivalent to the amount of pixels that are separating these two lines. And in order to clarify these two, this point here, we set the noise to 0, and that's also an assumption. But this is sort of just to characterize the actual function between, or the relationship between spatial resolution and radial position. And since the previous image was a little edge function that sort of elucidates the lines that were present in that inverse radon transform, and essentially this helps us pinpoint the exact distance at which the two lines converge, which indicates the exact radial coordinates where we experience a pixel separation that's equal to the amount from the original image. So we went ahead and took each of these points here, and then we plotted them. We plotted their radial position against the corresponding spatial resolution to generate a function between spatial resolution and radial position. And here's our simulation results. We generated the overall function between spatial resolution and radial position. We have two segments here. On the left side here, we have a constant section. This denotes the safe zone in which we have adequate sampling. And in the safe zone, we have a maximal spatial resolution that's calculated to be around 4 pixels. And then anything beyond that safe zone is going to be undersampled, and this occurs starting from 29 pixels away from the isocenter. As we expected, we observed a strong linear relationship in the undersampled region, And we also observe a constant max spatial resolution in the adequately sampled region. And one thing to note here is that the spatial resolution likely worsens linearly, and this is due to those gaps in delta S being increasing in magnitude that's corresponding to the radial position of at which they're taken. So overall, we assume that there was sort of like this this distinction between the adequately sampled region and the undersampled region, where we have a constant linear section here, and we have a constantly increasing spatial resolution as we approach the undersampled region. So just to conclude, we have some key points to go over. So one main point is that the radial aliasing threshold increases as sampling rate increases. And this creates a circular region in which the spatial frequency is maximized and we experience adequate sampling. And also we have a linear rate of decrease in spatial resolution as radial position increases beyond the aliasing threshold. Some future directions for to further explore this topic would be to run some simulations and observe changes from different sampling rates, reconstruction algorithms, and noise levels, and also being able to generate point spread function and the modulation transfer function from these images for improved characterization and robustness of the model. So those would be some extensions of our project that we could consider. So that's about it for us, and thank you for your attention, and we're happy to take any questions.
Oh, that's terrific. I like your simulation better than the one I use in class, actually, which has like the noise looking reasonable inside the circle, and then the outside stuff's all correlated. But this one's really nice, the way you see how everything gets blurred together as you go out radially with those two parallel lines. Makes it quite simple. Any questions from the class? No? Okay. Do you think in terms of the parallel lines and making your measurement with respect to those parallel lines, is there any higher order if we go one back? So here you've thresholded and you've said, okay, now my edge is below a certain threshold. But if you draw a perpendicular profile through the parallel lines as you move out radially, it's obviously that you get a complex function, right? It's not just a blur, it's some kind of interfering complex function, right? So it might be interesting to actually, characterize what the heck that function is.
Mm-hmm. And I think that's just sort of due to like as we approach the values where delta S is actually like equal to or like more than twice as great as delta L, we see like the doubled signal, right? So,
right. So you get sort of it'll be like a repeating pattern as you go out farther and farther in space. That's cool. I like this example. That's great. Okay. Any other things? Thanks very much for doing that. Did you send the MATLAB code for doing that?
I think so, yeah.
Okay, perfect. Thanks. I might put that in the lecture next year, actually. This demo. All right, great. Thanks. I think we have one more.
Hello, everyone. Can everyone see my screen and hear me okay?
Yeah, we got
Thank you. Yep, I am Jamin, and here's my colleague Abe, and we are going to present today on the X-ray instrumentation and some applications.
Just a minute, I'm going to pause because it looks like my microphone died. I'm going to get another microphone. Is this good? No, it's okay. All right, let's turn you up a bit. Okay, try again.
Sure. Can you hear me okay now?
Yeah.
Okay. So me and my partner Abe here, today we are going to talk on the X-ray instrumentation and applications in fluoroscopy. And this is the content at a glance. I will briefly talk about the principle of the X-ray that Professor McVie already taught us in the class. And then we'll go to the instrumentation of digital X-ray, TXR, which is the upright unit. We'll have some analysis of the radiographs. And at the end, we'll talk about some X-ray fluoroscopy. So once again, this is the principle of the X-ray. The X-ray tube is encased in the lead so that the radiation is not coming out in all direction. And the power supply is provided to the cathode over here, which generates the electron beam. And under the vacuum, there is an anode rotating over the motor, which is the tungsten in most of the cases. And then the X-ray beam is generated, and then there's a pre-filter which filters out the low energy radiation, and then the X-ray beam is projected over the patient or the object that we are analyzing. Figure over here, you can see the person standing, and then to keep in mind, when the X-rays are projected, the shadow is casted over the detector. Okay. So once again, this figure, this radiograph, Professor McVie showed us in lecture 2, I think, and he asked us which hand it is it left or right? So I would just ask you guys to guess this and give your answer in the chat, whoever are joining over the Zoom, and then we'll talk about it at the end. Okay, so this is the instrument here, the TXR upright instrument, and I'll talk more about the each individual parts. So this is the X-ray tube, which is, you can see here. This is the controller here for the X-ray tube to move it up and down and front and back. And over here is the collimator, which helps to regulate the radiation scatter. This is the front of the X-ray tube. This is how this the front looks for this particular XTR instrument. Here is the detector grid, which is 17 by 14 inch grid for this instrument. And I would like to talk a little bit more about this system console, which is usually on the other side of the leaded wall, and then the technician is operating this. So as you can see here, there's a power button to turn on and turn off the access system, but also there are some buttons here that you can use if the patient is pediatric, then you can use certain settings. And if the patient is big and tall, you can use certain different settings, right? So also to remember that clinically, it is very important as a first class of action that you need to take a proper X-ray or CT, right, with the proper settings so your images are better, and then you don't have to retake them and then give inconvenience to patients and avoid unnecessary radiations. So when you select these settings, the pre-settings, and also here you can choose some different settings about the chest, the leg, the skull, what X-ray you are trying to take, all these milliamperes, and then the voltage peak settings will updated, and accordingly the X-ray will be the radiation will be projected. And then the green button you can click to take the X-ray, and here is also the remote that the technician can click on it, and then the X-ray will be captured. Okay. So quickly, I'll just show you about the instrumentation. So as you can see here, I'm the patient standing here, and then this is the upright instrument. This one button helps you to move the X-ray tube up, down, backwards, and forward. So that's very easy to set the X-ray tube for the desired radiograph that you want. As you can see, the crosshair here with the laser helps you to get the center of your radiograph that you are intending to capture. And then later you can adjust your grid behind it. So then that becomes easier. And these two knobs here, as you can see, there's a light over me and then behind the detector. So that is actually help you to regulate the scatter of the X-ray, right? So you can avoid the unnecessary radiation, and then that way your contrast and the resolution of the X-ray will be perfect. The person goes behind the leaded wall and then clicks the button, and then the X-ray radiograph is captured. Okay. So this is the scan, and again, this is my scan that I got a couple years ago. So as you can see, and remember that I was facing in front of the X-ray tube, so my anterior part of my body was in front of the X-ray tube, and that the detector had my posterior part. So the X-ray, the radiograph that we are looking at is anterior to posterior, and also we are looking at the radiograph from an anterior to posterior view, right? So you can see here, my right side over here is the right side, and I'll just quickly talk more about the attenuation coefficients that we learned in the class. So the air over here is having the lowest attenuation versus the soft tissue, and the muscle in my neck area has a little bit higher attenuation. The lung you can see is pretty much darker because there's a lot of air inside. The bone for my C spine, cervical spine, it's scattering more radiations. You see it's more visually white. And if you can see a little bit more over here compared to my other teeth, there is a little bit of sharper white color in this area. That's because of the gold in one of my crown. So that attenuates more X-rays, right? Over here, we have the lateral view of the another X-ray. So just to give a little bit background, I was standing with my left side towards the exposure and my right side was towards the detector. And then again, you can see the crown which I have in my right lower jaw is visible over here. And there's a medical condition here. It's who are interested. It's a C cervical kyphosis, forward bending posture because of too much of social media and screen time, very popular these days. And then again, after a few months, that was corrected, and we can see here the cervical spine was corrected properly. Okay.
All right, then. So I'll be going over the applications of X-ray imaging in the application of fluoroscopy. So the principles of fluoroscopy are very similar with still image X-ray radiography, except in this case, we're pretty much taking a video instead of a single image. So the use of fluoroscopy is in order to observe dynamic processes inside of the body in order to diagnose disorders and guide surgical procedures. And this is typically done with the use of contrast agents. And these contrast agents are high attenuating. In this case, it would be stuff like barium or iodine. So over here, we see common procedures that use fluoroscopy as part of the workflow. And that includes in Figure 2, this is the barium swallow, which is where a patient is asked to swallow a cup of barium sulfate solution. And this allows the doctor to observe any blockages or any structural abnormalities in the esophagus of the patient. In Figure 1 over here, we see what we call a facet joint injection. And what is done here is that the joints are wearing away and they cause a lot of pain. So what happens is that the doctor injects the joint with a iodine contrast agent. And this allows the doctor to visualize, oh, the needle is inside of the facet joint. So they're able to inject painkillers into the joint itself and relieve pain. So there are two main dominating systems for fluoroscopy systems, and they're the flat panel detector systems and the image intensifier systems. I'll be starting by talking about the image intensifier system, which is over here. This is an older system design, and over here we see on the left is what the system looks like. At C over here, we see the X-ray tube. Oh, whoopsies. And B is the image intensifier column, and A is where the video camera is. How this works is that the system collects electrons through an input phosphor at the bottom of the column. And this phosphor converts the collected electrons into light. And this light is then passed into a photocathode layer. And this photocathode then emits electrons into the column. And these electrons are accelerated and focused by electrodes over here onto a output phosphor plate over here. The electrons are collected and then captured by the TV camera. And how it works is that since we're projecting electrons onto a smaller area phosphor, we get a much brighter image. This allows the image intensifier systems to have high spatial resolutions at very low FOVs. So the advantages to the image intensifier system is that they're less expensive than flat panel detectors. However, their spatial resolution, although very high at low FOVs, is limited by the field of view because once you expand it, you start losing the spatial resolution, and you're also limited by the TV system that you're capturing with. Because of this column focusing mechanism, the image intensifier systems also suffer from spatial distortions and defocusing effects. So due to advancements in a solid state electronics, we now have the flat panel detector systems. Over here, we see the setup over here. B is the X-ray tube and A is the flat panel image receptor. The receptor contains an array of detector elements. Over here, we see that there is a layer of cesium iodide, which is the input phosphor. And this converts the captured electrons into light, which then hit a photodiode array underneath the scintillation layer. And this converts the light into electrical signals. And that allows each of these arrays to represent an intensity of X-rays that is detected by the detector element. So because the flat panel detectors don't require a video camera, they just require they don't need a video camera, so they can be able to convert the electrical signals through a photodiode. Because of this mechanism, they also do not suffer from spatial distortions or defocusing effects caused by the focusing mechanism of the image intensifier system. However, they are typically more expensive, and they do suffer from low spatial resolution at very high and very low FOVs.
All right. So I don't know if anybody guessed the left or right hand, but if I'm the radiologist, whether I'm beginner or the professional, I might not guess whether it's right hand or left hand. That's my opinion. And that's why there is significance of the radio opaque markers, right? Usually, as you can see that for this photograph over here, we have this little lead sign which says R inside, and then also the markers has the technician's initials on it, right? So that you know that which hand is which hand, and especially if you don't have any knowledge how the X-ray was radiographed, whether it was the hand was facing the posterior or the dorsal side versus the palmar side, right, to the detector, it becomes very challenging to figure out that which actual hand is this. So I would recommend to go to the technician to go back to the notes, and worst case maybe repeat the radiograph for the patient. I'd like to thank you, Dr. McVie and the team, and Dr. Sean Miller for the X-ray videography. And if there are any questions, we'll be happy to answer.
Great. Terrific. Any questions from the students? We're minus 1 minute here, but one quick question. You guys have a particular interest in that X-ray system we were looking at for some reason, or?
Oh yes, yeah. So just because it's one of the system which is manufactured in US, and it's widely available to the offices like physical therapists, chiropractors, and things like that. So that's why we chose particular that system.
I see. So it's in widespread use. Okay, great. Well, thanks for the description of the digital detector and everything. We didn't really go into that in the class, so that was terrific. The students got that. Thanks everybody for the good presentations. And